Good morning, good afternoon, or good evening. My name's Joe McCracken, and it's my pleasure to introduce you to Acepodia, a clinical stage company that's working to make cellular therapies accessible to all patients with cancer. So Acepodia was established by Dr. Sunny Shao, who earned his PhD from the University of California at Berkeley, and who has spent a lifetime studying the innate immune system and cell biology. Together with Dr. Pat Yang, they decided to establish Acepodia to fulfill the promise of cellular therapy for all patients. Dr. Pat Yang was the head of manufacturing sciences at Merck, at Genentech, at Roche, and later at Juno Therapeutics. Our company also had some recent additions. Dr. Mark Gilbert from Juno Therapeutics is our Executive Vice President of R&D, and Dr. Thorsten Graf recently joined us from AbbVie, where he was the head of early stage oncology research. Our headquarters are located in the San Francisco Bay Area of California, and all of our clinical trials are being conducted in the United States, most notably at the MD Anderson Cancer Center and also at the Northwestern Medical Center in Illinois. So we only have a few minutes to introduce you to Acepodia today, so I just want to make three key points. One, our unique effector cells leverage the strengths of the innate immune system with enhanced anti-tumor activity. Two, the potency of our effector cells is then amplified by our unique targeting technology. And thirdly, our process enables access to all patients with hematological and solid tumors. So let me talk a little bit about the innate immune system. The key strengths of the innate immune system are that cells transferred have a low risk of graft versus host disease. They have a low risk of cytokine release syndrome and a low risk of neurotoxicity, all challenges with cells of the CAR T family. These cells are also appropriate for allogeneic or off-the-shelf uses. At Acepodia, we're focused on two cell types of the innate immune system: NK cells, which kill abnormal cells without the need for prior immunization, and their killing capacity is not restricted by the target cell's need to express the major histocompatibility complex, or MHC. We're also working with gamma delta T cells, which attack target cells directly through their cytotoxic activity or indirectly through the activation of other immune cells with a broad range of antigen recognition ability. But not all cells of the innate immune system are created equally. At Acepodia, we select and then work with rare subpopulations of NK or gamma delta T cells based on their increased activation receptors and decreased inhibitory receptors. In the middle panel, you can see our proprietary NK cells identified as ONK compared to the NK92 cell line. And you can see the dramatic increase in expression of activation receptors like CD16, NKG2D, and other activation receptors. And you can also see the decreased expression of inhibitory receptors like NKG2A, PD1, TIGIT, and KERS. And the same is true for our gamma delta T cells. But these cells of the innate immune system don't inherently or naturally target tumors. They have to be directed or targeted to tumors in order to maximize their activity. At Acepodia, we use a proprietary chemical method employing DNA hybridization that enables us to link more than 100,000 antibodies to the activation receptors on the surface of each NK cell. This increases the avidity of the interaction between our antibody cell conjugates and the tumor dramatically, almost like Velcro with so many points of contact. Our cell conjugation method also unlocks multiple NK activation receptor pathways, such as CD16, LFA1, and the CD2 family. And importantly, we can conjugate any antibody to any effector cell using our proprietary technology. So how does this address some of the challenges of the first generation of cellular therapies? First, our proprietary potent NK and gamma delta T cells can traffic to tumor locations where they are concentrated and can directly kill tumors. Second, our antibody cell conjugate links more than 100,000 antibodies to the effector cells, which increases their avidity against low expression tumor antigens and also can prevent tumor escape. Thirdly, the cell activation when our antibody conjugates interact with the tumor overwrites inhibition signals from the tumor cells. And importantly, this superior potency results in activation of the adaptive immune system. So in the panel, in the lower panel on the left, you can see data demonstrating that one of our compounds, A1702, which is a antibody cell conjugate directed at the HER2 tumor antigen. In the left panel, you can see that our antibody cell conjugate migrates and is concentrated at the tumor cells. In the middle panel, you can see that our antibody cell conjugate results in the production of interferon gamma when our cell conjugate engages with target tumor cells. And in the panel on the right, you can see that our antibody cell conjugate 1702 recruits T cells once activated. Importantly, our cell platform is adaptable to any targeting antibody or effector cell. So we can attach multiple antibodies targeting different antigens to the same effector cell. And for us, cryopreservation is key to our off-the-shelf products. So for us, we won't take a product forward unless we know that it can be cryopreserved and shipped to patients anywhere in the world for clinical trials or in commercial use. Our technology is scalable, it's reliable, it's cost-effective, and it's safe, and has the promise of making our cell therapies available to patients anywhere in the world. So looking briefly at our company's product pipeline, you can see A1702, which is an antibody cell conjugate, an NK antibody cell conjugate that targets HER2 expressing solid tumors. Notably, we can treat patients that only express HER2 at the 2+ level, as well as the 3+ level. We are currently in our phase 1 clinical trials, and we expect to have our data from our phase 1 study completed in the second half of this year. Our second leading clinical stage project is ACE1831, which is a gamma delta T cell that targets the CD20 antigen, and we expect to file our IND shortly and in the second half of this year. Importantly, we're currently in discussions with a number of multinational pharmaceutical companies who are very interested in utilizing their novel antibodies or binding proteins in our antibody cell conjugate platform. So just as an example, we're working with a multinational company who has 2 antibodies, one for a target that they were quite fond of, and they tried to develop as an antibody drug conjugate. Unfortunately, they couldn't develop an ADC that was properly internalized, so they gave up. Another antigen was expressed in very, very low density on the surface of tumor cells, but was very specific. And again, they were unable to develop an ADC that was of adequate potency. So we're evaluating those antibodies as antibody cell conjugates and developing some very exciting data. Important, because of the enormous efficiency of our technology, we can actually go from an idea to an IND in 6 months for precedented antibodies. So we'd be happy to talk to you about your antibodies. If you're interested in working with us and adapting them to our antibody cell conjugates, and our contact information is here. And with that, I would just like to thank you for your time and for your attention on behalf of all of my colleagues at Acepodia.